Dun 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 dun. Digga 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 dun dun. Ba-ba-da-ba. The miracle of life is a concept you are all probably familiar with. Male and female gametes come together and each donate half of their genetic code. But how does a male's genetic code turn into a life-giving package? The answer is spermatogenesis. Spermatogenesis is a process of producing sperm where the goal is to produce a continuous supply of mature spermatogonia that provides genetic diversity to any and all offspring. The main site of spermatogenesis is the testes, but signals from other parts of the body, such as the brain, are also necessary. The process of spermatogenesis begins in puberty. Through puberty, the frequency of gonadotropin-releasing hormone, or GnRH, begins to increase and reaches a point where there is enough being produced to stimulate spermatogenesis. Spermatogenesis will continue as long as there is a sustained high-frequency tonic release of GnRH. It starts with hormone signals from the brain, which travel to the seminiferous tubules of the testes and stimulate the development of germ cells into spermatozoa. The first of these signals is GnRH. This travels to the pituitary gland where it will stimulate LH and FSH, or luteinizing hormone and follicle-stimulating hormone. These are the two main hormones of spermatogenesis. Once LH and FSH have reached the testes, FSH targets Sertoli cells. These are an important component in the blood-testes barrier. The blood-testes barrier is comprised of Sertoli cells and junctional complexes that protect the developing haploid sperm cells from any agents in the blood that might cause harm. Without this, the body would attack itself and destroy the haploid cells that register as foreign to the body. LH targets Leydig cells, which produce testosterone. Testosterone does many things in the male. Most importantly, testosterone supports spermatogenesis and accessory sex gland secretion. Testosterone also acts as a substrate for estradiol and dihydrotestosterone, which allow for the development of secondary sex characteristics and sexual behaviors that lead to mating. Both FSH and LH are required for adequate testes function. Once these signals reach the seminiferous tubules in appropriate amounts, spermatogonia, or the germ cells, begin to develop through phases of proliferation, meiosis, and differentiation. Proliferation is a series of mitotic divisions which occur during the fetal life, as well as of the rest of the adult's fertile life. Mitosis is a cell division that results in two daughter cells with the same number of chromosomes as the parent cell. The spermatogonia then undergo meiotic divisions that produce haploid spermatids. Meiosis has two phases, both of which include a prophase, metaphase, anaphase, and telophase cytokinesis. In phase 1, the homologous chromosomes will separate. Once the homologous chromosomes have separated, they will move on to meiosis 2, which is where the sister chromatids separate, resulting in 4 haploid daughter cells. These spermatids then develop further during the process of differentiation. The process of differentiation has 4 phases: Golgi, cap, acrosomal, and maturation. In the Golgi phase, the acrosome is formed from the Golgi complex. Then, the centrioles will begin to migrate and soon will form the neck, which is the attachment point of the tail and where the annexosemae or flagella will begin to elongate. In the next phase, or the cap phase, the acrosome is stretched over the nucleus, forming a cap-like structure. In this phase, the flagella will also begin to elongate from what used to be the centrioles. In the acrosomal phase, the nucleus begins to elongate. Here, the artist accidentally absorbs the neck into the nucleus. Silly artist. That does not happen. Instead, the neck becomes fully formed and the flagella elongates even more. Finally, in the maturation phase, the mitochondria will migrate towards the neck area. They will form a spiral assembly around the flagellum that will power the spermatozoon's movement. This produces viable spermatozoa and thus ends the spermatogenesis process. As the spermatozoa mature, they move from the blood-testes barrier to the peripheral and luminal compartment. A single cycle of spermatogenesis has 8 stages, which means there are 8 different types of cells found in the seminiferous tubules at any one time. The 4 categories of germ cells are spermatogonia A, I, and B, primary and secondary spermatocytes, immature spermatids, and mature spermatids. This cycle results in the daily release of sperm, even though in the bovine species it takes 61 days to produce a single mature spermatid. The ability of domestic livestock species to produce and release billions of mature sperm each day has to do with the length of the seminiferous tubules. The tubules are so long and thin that the sperm are constantly being released somewhere along the tubules in a cyclic fashion. Once the cells differentiate during a process called spermatogenesis, they are mature and can be released. The releasing process is called spermiation. Once released, mature sperm are delivered to the lumen of the seminiferous tubules and then to the head of the epididymis for storage prior to ejaculation. I will now slowly write out the word cryopreservation so that you know we are no longer talking about spermatogenesis, but rather cryopreservation. Cryopreservation is used when livestock producers wish to save ejaculate from their sires for later use in artificial insemination. Cryopreservation is a process by which spermatozoa can be frozen, usually in sperm straws, stored for later use, and then thawed when they want to. This is particularly popular for bulls who express desirable characteristics such as carcass quality, health, or temperament. Maintaining quality of sperm is key in this process. Spermatozoa quality is assessed by percentage of motile sperm, progressive motility, normal morphology, intact cell membrane, and the ability to survive osmotic challenges. If the spermatozoa fail these assessments, they may not be able to fertilize an oocyte. There are 2 main categories for sperm that do not pass: primary and secondary abnormalities. Primary abnormalities will not be able to fertilize, while secondary abnormalities have a chance of successful fertilization. Abnormal sperm will be discarded unless the male has a trait that is more valuable than the potential cost of the defect. If the sample is discarded, another will be taken. In the case of the bull, it is typical for him to be ejaculated several times over a few days before the first sample is collected. This helps decrease the amount of abnormalities present in the collected sample. Cryoprotectants are used to help protect cells from cold damage and minimize the formation of ice crystals within the cell membrane. These can interfere with the quality of the sperm and the ability to successfully fertilize the oocyte. The most common cryoprotectant used is glycerol, with nutrients such as fructose and glucose to maintain the cell's metabolism during cryopreservation. Other cryoprotectants, such as cow's milk and egg yolk, are used in seminal extenders, as well as providing macromolecules that minimize cold damage and provide nutrients. Seminal extenders also include pH buffers, energy sources, and antibiotics. One method of cryopreservation is vitrification, a process that uses concentrated solute to freeze the samples without producing ice crystals. Since the concentrated solute medium prevents the development of crystals, vitrification can be performed quickly, thus preserving the quality and integrity of the sample. Even with careful vitrification and use of cryoprotectants, about 50 of the sperm will be damaged during the freezing process, leaving you with only 50% to fertilize with. Frozen semen is often used in goats, cows, and sheep undergoing genetic improvement programs, accelerated herd replacement programs, or elimination of congenital conditions. The most common of these is genetic improvement. Though fertilization and insemination is a complex process that may seem miraculous, it can be broken down and understood starting with the steps of spermatogenesis. Hormonal cascades stimulate the production of sperm when GnRH triggers FSH and LH during puberty in the male. These hormones target specialized Sertoli and Leydig cells to encourage growth of crucial structures and secretion of testosterone. The hormone that supports spermatogenesis and accessory sex organs is testosterone. Spermatogonia undergo a differentiation process involving mitotic, meiotic divisions, and ultimately differentiation that creates the spermatid. And that is the process of spermatogenesis.